Yes.
Yeah. Thank you very much. Yes, I have been working at the Department of Anatomy, so I have been teaching cell biology, developmental biology, and this kind of thing. But I'm biologist. And my presentation is dealing with human beauty. But I don't consider who is beautiful and who is not. My starting point is our cells are beautiful, all animals and organisms are beautiful, so we are beautiful. We can discuss it later. But in the end of my talk, I will consider the aspect of aging. Because I know that the only thing I see actually of you now at the moment is your face. You are just covered. The rest of your body. Face is the critical. And the past professor of anatomy said that people are never happy and satisfied, or at least completely satisfied with their body. And I think that's the point. And he also continued and said something, whatever I do and make my best, it's never enough. So we are never happy. We are never happy with our body. It's my one point, but I don't discuss about that. So I will start my talk, and let's see how it goes. In the end, I have one video. I have made some recordings from, I don't say primitive animals, but tiny microscopic animals, and I will show the video in the end of my talk. So, cells in our beautiful and aging body. That's the starting point. And that's at least a part of our body. That's one of our friends. And there's an artistic view of human body. They are quite similar. The artist has been very successful. And of course, when we are now moving from atoms to molecules, biologists need only molecules. We don't consider, we are not interested about atoms because we don't see them. So molecules and how they organize, for instance, into DNA, and from DNA to cells, cells into tissues, and tissues into individuals. And I took DNA here because we are in sokeri kulma. And sugar, sokeri, is one key molecule in DNA. Ribose, DNA, deoxyribose nucleotide, DNA, or RNA ribonucleotide. So ribose, sugar is one key molecule in DNA. That's why it's nice we are here. But it's ribose. Glucose we need for ATP synthesis to get energy for cells. But sugar is one key molecule also in DNA. Ribose is less stable. That's why RNA is very, very easily breaking down. But deoxyribose in DNA is more stable, and that's why we find DNA from fossils and so on. It's very different models. And now I come to, we were discussing lunch break about linear. The way we think about evolution is normally linear. We were discussing what of that. Linear, that first there were fish, and then frogs, and then crocodiles, and apes, and humans. I just did this presentation just to remind you, or at least give some ideas for discussions, that maybe there are also other ways to think of evolution. Because I think traditional idea, even when the Human Genome Project got ready about 20 years ago, then we first realized the first time And the gene number in humans is not the highest in the whole living universe or organism. No. It was a little bit disappointment for humans, because we are the kings. But actually we are not. I don't know, can you see from there in background, number of genes in humans, 25,000 about. Some say that 29,000, but no matter, about that size. Mouse, 25. Chicken, 23. Frog, 28. Sad. Very bad. I don't like that. And some fish, 31,000, and so on, so on, so on. And even Drosophila, 14,000 genes. The small fly flying in our kitchen at the moment. We don't like them, but they have plenty of genes and they are beautiful. So they have everything we have. They have about the same cells also, of course. So, but there's one interesting echinoderms, sea stars and these kind of animals. We think they are very primitive. And we want to avoid them when they are swimming in Atlantic. Just they have quite a nice number of genes. So we are nothing special in that sense in the evolution. We are a compromise. And talked about the number of chromosomes. The term chromosome comes from that in early histology and microscopical studies of our heredity, chromosomes were easily stainable. Chrome color chromosomes, stainable particles. And if we think about Homo sapiens, 46 chromosomes, potato. And from that, the potato is beautiful, almost the same. So we are beautiful, and potatoes are beautiful. But they have even more genes and chromosomes. What about the simple fish, if I can say? Now, it's maybe good to say, not to say simple, but fish, because they have a huge number of chromosomes. Dogs have even more than we. So, we are eating chromosomes. We are eating huge number of genes when we are eating something alive. Cat and mouse, maybe they are friends because they have about the same number of genes. No. Okay, but strange. The bird you are going for fishing, just nothing actually very surprising. We have to respect also that animal. And even when we are baking or brewing beer, surprisingly high number of genes, but plants are strange. Even this Getoneidonlaukka, grown also in Finland, plants multiply their chromosomes, their genes, and so they may have huge number of genes. So my message is that we have to respect all the animals, animal forms, and not just to say that they are more primitive. They are different. But my topic was also about the cells. So the cells are about the same we have, but they are organized in different ways. Sea star, beautiful animal. That's the dead one, but as alive. Same number of chromosomes we have and about the same number of genes we have, but they live about even more, maybe 100, 150 years and so on. So, frogs. The sea star has 5 arms. Frog has, if I add the drawing here, also we humans have 5 arms. Can you count them? Head, neck, and hands and legs. 5. star here? Yeah. Oh, nice. But why 5? I don't know. Why 5? So we need 5 fingers, 5 tools. You may try walking. When you walk, you can feel that you need big tool for walking. It's not the last tool you touch the ground. If you don't have the big tool, walking is very difficult. And of course, thumb is just important for taking the tip in a pen or whatever. So this is from our home. It was.
It is and
It is something here. Now it's coming.
No, it's gone.
okay. Okay.
Okay. We need 5 fingers at least for many things. This picture is out of focus that you don't see what kind of pizzas my wife is making. But 5 fingers is extremely important for everything we do. We can continue the story, but 5 is in C stars involve fingers and hands. One strange number. I don't know what one would physics say about number 5.
Yeah,
42. Okay. But, okay, our basic organization resembles many animals. But do we go through metamorphosis, change of form, or do we mold Gorilla? I don't know. Maybe. If I think that it's metamorphosis is very common with insects. They have larva, pupa, and larva. And if we think about humans, this is a human fetus about 9 weeks old, and 16 weeks, and newborn, and so. Do we go through metamorphosis? Of course, in biological terms, we do not. But it looks like we would go through a kind of metamorphosis. And we also mold. At birth, we get rid of fetal membranes, amnion, chorion, the membranes that surround us. So, evolution is conservative. It doesn't create so much big new inventions. We just use them in different ways. The head in fetus is 1/3 of the body size, but then the relationship changes quite a lot. And I added this sexual differentiation because in that leaflet there was a picture about bisexual stage of human embryo or fetus. And it doesn't consider only genitals, I will show later. The mammary gland is also an interesting organ. We have so-called mammary gland line in our fetal body. And the text here now is saying that the female pattern is dominating. And that means that every individual, human individual, would be female if he would not have Y chromosome, or even though he would have Y chromosome, but if it does not produce testes, the organ testis that produces androgens. In any case, the human phenotype would be female-like. And we know even that the estrogen receptor is evolutionary older than androgen receptor. So the male phenotype is younger than female. So Adam and Eve complexes. So we were ambisexual during embryonic development and partly fetal. We all sexes developed. And these organs and items develop also in the boy fetuses, but androgens that come from testes regress them away. Only the nipples are left, but the gland, the milk-producing tissue is regressed by male androgens produced by fetal testes. So if the fetus has testes, the male phenotype will be expressed. Otherwise, the female phenotype. So this also shows that we are somehow related to other animals also, like a lady pig. They have many mammary glands. We don't, but we have the small rudiments for putative development. So, what about, this is the picture you have seen in many places, but the boys and guys who draw these kind of pictures in toilet walls, they don't know actually that they are drawing the picture of their genital organs during fetal development. And this is again to remind us that the female pattern is the original pattern. This is the female indifferent gona. This is the, I just painted some structures of this. This is the male pattern and this is the female pattern. And you see the female pattern in adult individual is about the same as the fetal pattern. But the male pattern takes a different pathway induced by testicular androgens the individual produces during pregnancy. So, what actually is this drawing means? That the clitoris in women, actually, the corresponding structure in males is man's penis, the tip of the penis. Small labia minora in ladies, women, the corresponding structure in males is the shaft of penis. And actually, what is surprising now and then, scrotum. Actually, the corresponding structure in females is labia majora. Single. These are the examples I just want to somehow bring together that nature is conservative. And the cells and tissues, the primordia are very often similar, but somehow they are guided into different developmental pathways, like the sexual differences. That's why sex is a complicated story, because it's not only male or female, it can be something between. That's the point. And so this still is to show that how these structures are in you differentiated under influence of fetal androgens. And why at the moment there are some worry about why boys may have more developmental abnormalities. Because this is the pathway that must be actively guided to proceed to male pattern. And if some disturbances, like environmental hormones, disturb this, then the male pattern may somehow disturb. That's why in the world we are more worried about the male fertility than female fertility. Because the female pattern is the dominant pattern. So, this picture we have in the program, this is just to remind us, this is a fetal ovary, female, and this duct will develop to oviduct and uterus, munajohdin kohta. This duct is regressive, and this is the male duct that would, if this would be testis, this duct would develop. But now it's disappearing. It would in males develop into epididymis, risaciles, and the ducts of male reproductive system. But now it disappears because fetal ovary does not produce any hormones. So the male pattern disappears and female pattern remains. That means ovida uterus development. It's interesting. Either or. And dual system. And now from that I come to the general pattern that as in our body we have right and left side, right and left hand or left toe. Cells also have, they have information about right and left. So it's not a new invention, it's already in cells. They are polarized. And this is just an introduction to that how old actually we could live. This is approaching the teen aging, going to the end of my talk. But this is the experimental approach. There are tortoise, Galapagos tortoise, Kilpicurna cells cultured on the dish. They can divide about 125 times. That would mean theoretical maximal age of 175 years. If human cells are cultured in similar conditions, they divide about 55 times the maximal division times. And it means for about 120 years of maximal age for humans. So our cells, our genes have programmed ourselves to die after a certain number of divisions. Finally, I come to the point. We have one type of cells at least that do not divide at all, and they are neurons. And that's interesting. But let's see. I hope this video will open. It's from Olympus company, but they have very nice videos about cell cultures and division. I hope it opens. Okay. It's going to take now 2 seconds. But this will show the phenomenon right and left handedness in cells. After division, you will see that when cell divides, the other cell goes in that direction, and the daughter cells into opposite direction, right and left. Let's see. I may start already. The nucleus is red. That cell will divide. It is getting round its shape soon. It will soon divide. Now it's soon. Okay. Daughter cells, and the other one goes there. The other one goes into opposite direction. So right and left handedness is programmed in our cells. So it's nothing new actually that we have bilateral symmetry in our body. Because cells are bilaterally symmetric. Plus and minus in that sense. Yeah? Okay. So, about aging. My wife had her 60th birthday a few weeks ago, and we invited our neighbors, and I used the opportunity and take some now I come to the phase, because that's the point we are never happy with, at least our own phase. So, our neighbors. You see, of course you see what I'm looking at, or pointing soon. We are all disappointed with these sprinkles. But this next labels I offer you to, you may have seen them somewhere else. there's a global terminology for different wrinkles. And if you want to make your life more easy and nice, you can give some certain names to your specific wrinkles. And this is just to summarize. Surprisingly, Sibelius frowns. We all have. If we don't yet have, you just wait for something. You will have that. Or bunny lines, Jänne, Pupujussi, and so on. Tier, kyynel, vako, kyynelkura. I had first year spelling mistake, there was cows feet. But luckily I noted that R is missing and it's varitsejaaka, crow's feet. Marionette lines, do you have? Yeah. And this is interesting, vertical lip lines. Now we come to sexual differences. This is typical for ladies, women.
Smoking ladies.
And non-smoking also, but let's see why. Why is that typical for women? It's because of cells. If I open that page from our virtual microscopy pages, this is a section. This is a modern microscopy. We don't use normal microscopes anymore. We scan the slides and make a virtual setting up. And this is like a Google map system. So we can focus into the specimen and see, for instance, here, the epithelium, the thickness of epithelium, the outer surface varies. And for instance, in the eyelids, epithelium is very thin. This is from eyelid area, and in eyelids we easily get wrinkles because the epithelium is thin. But what is also changing during aging is the collagen fibers that form the numerous huge packets of elastic fiber under the skin, and during aging and maybe due to UV irradiation, elasticity is gradually decreasing, and you'll see the results. Yeah. So that's, and why men? More hair, less wrinkles. In hair, there is also talinauhanen, the sebacous gland, but hair is like it's like a stick supporting the crown or skin. Okay? Our next neighbors, you don't see, you can say, of course, I don't see the wrinkles, but he has. But I took the Pekka, who is also living in our neighborhood. You see, Pekka is about 10 years older than he, but you see the lip, upper lip is nice in that sense. Okay, he's man, and there is a very thick hair follicles in upper lip of men. So, just require more hair in your upper lip. Okay, what about this? This is my forehead, the neighbor. Not only the tissue, but why we have the almost similar wrinkles in our every face. It's maybe due to the muscle tissue under the skin. And I took a picture from anatomical model. The tension of muscle tissue also decreases by aging, and it just wrinkles. It gets looser, and so. And around lips, we have this circle muscle tissue, and when they are just losing their elasticity, it helps us to get more and more wrinkles. So, it's in our body, it's also in the muscle tissue. Not only just that we can use creams and so, but something happens also under the skin, in the muscle tissue. So, this is aging. The cells are aging because they don't divide anymore so actively, because there is a limit for divisions for cells. Okay, but this is, actually I took this picture of myself, so sorry for that it's from myself, but I didn't want to ask my neighbor or my wife to come, because I would like to serve. Because you said that in quantum physics, or it's not possible for people to be in two places simultaneously. But in biological terms, we can. This picture I took from the mirror. And when you go and look your face in the mirror, you think that, who is that person? But you remember in your head, it's me. And you feel, I'm not so old, I am much younger. So, in memories, we can be in two places. Can we? I think this is the, do you recognize this feeling?
Yes.
Yes.
Yeah. Yeah. Something similar, only the foreheads. About you recognize. Okay. So, I come to the brains, and this brain issue is a bit broken. It's from our teaching materials, but now I come to the point again about the cell divisions. Neurons, which form basic structure of our brains, they are regular cells. They don't divide once they have differentiated. So when you experience something, the neuron will store it permanently until you die. That's why we have that experience of my childhood, and I feel my mentally younger than I am externally. Neurons have saved that for information and our identity. And if we think the brain tissue, we have huge of neurons because the cortex of brain is forming this kind of folding. We have numerous neurons, we have numerous capacity to store information. Also forget, but store. That's the main cause. Neurons, we can stain them and see that really there are a number of cells. And what is specific for neurons? Their size, they are the biggest cells in man's body, but they are the second biggest in woman's body, because oocyte is a little bit bigger than the biggest neurons. So they are huge cells, they have enormous capacity to make learning and these kind of things possible. That's this small picture, unfortunately, is too small. But what means Alzheimer? Actually, it means that these neurons start to regress, and we will lose neurons, and that means we lose memories. In that sense, it's quite simple. And now I would like to show some videos I have just at home made from the water of Baltic Sea, from south, quite near here. And I made these videos, that's why I'm quite happy with this. The quality is not the best. This is made under microscope, so focusing is not easy because they are moving, the animals. This first is algae, Pilevä. It's a one cell is like an arrow or tree. There are at least 1, 2, 3, 4 cells. And this is the Sinilevä, that poisonous or more or less poisonous. It's a different algae. But I kept radio behind open when I made this movie. I didn't select the music. And with these movies, I would like to propose you one idea for discussion. Because what I want to tell is written here. What did the video recording of microbiology and rotifer animals teach to me? At least I experienced that. Many of basic skills and capabilities, like musicality and sense of rhythm, are inborn properties of ourselves. I don't say is it so, but we may discuss, and I close this presentation and let's look this video. There are three small. Those are single cells, but they are joined with each other and moving in relation. This is a rotifer animal without music. But just look at the rhythmic movements and ciliary movement. That kind of cells we have also in our body.
Onko se ripsi vai rataseläin?
It's a one maybe 100 cells or so. It has a stomach, it has brains, it has eyes, spots, it has everything to be happy. This is with the same animal with the leg support and with the music. You may say this was accident or my imagination that cells have a sense of some musicality or rhythm. But I think that we could not be musical or even speak without ourselves. So the property is in ourselves somehow.